Online classes. Today we'll discuss chapter 2 of class 12 biology, sexual reproduction in flowering plants. This is the fifth session and the topic of discussion is artificial hybridization and double fertilization. These are the 7 different sessions. Already you might have seen 4th session, and please complete all the 7 sessions to complete different learning units. At the end of the lesson you will be able to understand and describe the process of artificial hybridization, to understand and appreciate the process of double fertilization, to understand and appreciate the significance of double fertilization. You are well accustomed to some of the old concepts like the anatomy of the flower, various terms associated with description of a typical flower, and you also know about pollination. You also know about the agents of pollination. So with this background, we will be discussing these topics. You can see what beautiful flowers. These are wild flowers growing near Depar Bell, a Ramsay site. Magnificent view. Our topic today is artificial hybridization. Let us try to define what is artificial hybridization. Artificial hybridization is defined as the process of crossing over two genetically different individuals having desirable traits to obtain an offspring having superior trait than the parents, and can be achieved by emasculation in plants. You can see that it involves certain stages, steps. Total 7 steps are there. Number 1, selection of parent, evaluation of parents, emasculation, bagging, tagging, pollination, harvesting and storage of F1 seeds. Let us now discuss these steps one by one. Selection of parent. The selection of parent mainly depends on objective of the breeding program. Besides the targeted breeding program, increased yield is always an objective of breeder. It is essential that all the characters which we want to improve should be present in one of the other or the other plant. The selection of parent is basic step in hybridization program, and often more than anything else determines its success or failure. The second step involves evaluation of parent. Generally, a variety well adapted to particular area or location is selected for hybridization. If the performance of parent in that area where breeding is to be done is not known, then evaluation becomes necessary. Evaluation of variety selected as parent in the breeding program for susceptibility to pathogen or disease present in the area need to be done, because in case evaluation is not done, then there may be transmission of those varieties which are non-resistant to pathogen, or even drought condition effect on the plants. So evaluation is a very, very important step for the process of artificial hybridization. Let us go to step number 3, emasculation. This is a procedure. If you split the word emasculation, E is removal, masculine is to male, means it's a procedure by which the male part of the flower is removed. Emasculation is essential to prevent self-fertilization in the flower of the female plant. It involves removal of stamen or anther of the flower without affecting the female reproductive organs, and that too, it should be done in immature flower. Emasculation may be carried out by killing of the pollen grains of the selected female parent. Once emasculation is done, then comes step number 4, that is bagging. It has got its significance. The name is suggestive. Immediately after emasculation, the flower or the inflorescence are enclosed in suitable bags, preferably butter paper bags, to prevent random cross pollination. We want target pollination. Random cross pollination is not a wanted thing. The bags are tied to the base of inflorescence or the stalk of the flower with the help of a thread, wire, or pins designated for the purpose. The bags can be removed usually 2 to 3 days after pollination, after the danger of cross pollination is over. So that is about the concept of bagging. It is simply a word that I would like to introduce is quarantine. Segregation of unknown source, unwanted source, so that we get a targeted experimental outcome. The fifth step is about tagging. Tagging is to tie. Tie what? A tag. The emasculated flowers are tagged just after bagging. In most of the crop, circular tags of about 3 cm diameter or rectangular tag of about 3 into 2 cm are commonly used. The following information is recorded on the tag with carbon pencil. There is a common pencil, HB pencil you can use. Date of emasculation, date of pollination, detail of cross, that is name of the female and male parent. The name of the female parent is written first and that of the male parent is written later. So this tag gives a fair idea about what type of crossbreeding experiment is being done. Then comes another very important aspect, that is pollination. We know pollination is transfer of pollen grain, but how? In case of experimental condition, it has to be done. The application of mature, fertile and viable pollen on the top of receptive stigma with the objective to carry out fertilization is known as pollination. For carrying out hybridization, it is a prerequisite that the pollen should be viable and stigma should be receptive. Generally, the fresh pollen from mature anther should be used, and at the time of anther dehiscence falls within the duration of stigma receptivity. The procedure consists of collecting pollen from freshly dehisced anther of the male parent, dusting this pollen into the stigma of the emasculated flower. Now, how this pollen transfer is done? The pollen grains are collected in petri dish or a bag and are used for dusting the stigma of the female inflorescence. Mature anthers are collected from the flower of male parent. The pollen is liberated on a clean petri dish, applied to the stigma with the help of a camel hairbrush or piece of cloth or paper or toothpick or forceps. So artificially, the target pollen is introduced to the stigma of the experimental flower. And then comes the most important thing, harvesting and storing of F1 seeds. Why it is to be done? Because once a crossbreeding experiment is done, we have to ensure that the outcome of the cross is successful or not, whether we are achieved our objective or not. That is why this step is important. Harvesting and storing of the F1 generation seed, the crossed heads or pods should be harvested and thrashed. The seed should be dried properly before storage. Hybridization has resulted in the development of new variety in almost every crop. This is the outcome of harvesting and storing. When we talk about a topic like artificial hybridization, there should be another thing that should be in our mind, that okay, what is the need, what is the importance, what is the significance? Let us talk about the significance. Artificial hybridization is a process in which only desired pollen grains are used for pollination and fertilization. So the advantage are they increase the yield. Two species combine to form the best of organism, eliminating the unwanted qualities of both the parent species. So we get a product better than both the parents. That is the significance of artificial hybridization. After this, let us move to the second part of the concept, double fertilization. Double fertilization is a characteristic of angiospermic plants, for flowering plants. In this process, two male gametes fuse with one female gamete, whereas one male gamete fertilizes the egg to form the zygote, whereas the other fuse with the two polar nuclei to form an endosperm. You remember that there are two male gametes, the first and the second. First one with the egg and the second one fused with the polar nuclei. So in the first there is production of a zygote, and in the second fertilization there is production of an endosperm. The first one results in the production of a zygote which is diploid, and the second one results in the production of fusion of three nucleus. So it is a triploid endosperm. Now, what is double fertilization? After entering one of the synergid, the pollen tube releases the two male gametes into the cytoplasm of the synergid. In previous topic, we have already done this. As soon as the pollen tube enters the synergid, the filiform apparatus guide the pollen tube, and then the male nucleus they are released for the process of fertilization. One of the male gamete moves towards the egg cell and fuse with its nucleus, thus completing the syngamy. The second male gamete moves towards the two polar nuclei located in the central cell, fuse with them to produce a triploid primary endosperm. In short, you call it PEN, and the nuclei is called PEC, primary endosperm cell. This involves the fusion of three haploid nuclei, and that is how we get the typical term triple fusion. Since two types of fusion, syngamy and triple fusion, take place in an embryo sac, the phenomena is termed as double fertilization. This is an event unique to the flowering plants only. The central cell after triple fusion becomes the primary endosperm cell PEC. The nucleus is called PEN, primary endosperm nucleus, and they develop into an endosperm while the zygote develops into the embryo later on. Now visualize this diagram. We already seen this diagram. You can see the pollen tube entering one of the synergid and guided by filiform apparatus, and then you can see that once it has gone inside, that pollen tube opens up and releases the male gamete. The first male gamete has moved towards the egg cell, as the second male gamete has moved towards the polar nuclei. With the egg cell they produce the zygote, and with the second one, that is polar nuclei, they produce the triploid endosperm cell. What after that? After that, you can see in this typical diagram, this is nothing else but embryo sac. It has 7 cells now, because the polar cell they are fused together, and this is 3N, triploid. This is the zygote, which is 2N. This zygote, how it develops into the embryo proper, and when the endosperm should grow. Please remember, endosperm production starts faster than that of embryo production. It is something like when some friend of yours visit your house, food items are already ready to be served. In case food item is not there, then there is nothing to offer to your friends. So this cell gets ready with the food. Endosperm is the nutritive tissue. It provides nutrition to the developing embryo. Now please see this diagram. In diagram number B, you can see that it divides into a large cell that is called the epibasal cell, and there is a hypobasal cell. Then the hypobasal cell is quite big, and the epibasal cell starts dividing around different planes, and over a period of time it produces a long chain of 8 cells. You call it suspensor, and at the base, because of horizontal and transverse type of cell division, it produces a round mass of cell, and this stage it is called as globular embryo. Now slowly over a period of time, you can see the suspensor has stopped dividing. 8 cells are there in the chain. This is important for pushing the embryo inside the developing endosperm. and at the base, the heart sept embryo, both the side there is differentiation and extension take place. Now it looks more like a heart, so the typical name heart sept embryo. And still on further development, you can see these two end of the heart sept embryo, it grows more and more and it forms the future cotyledon. And in between these two cotyledon, there is the embryo. You can see in the diagram the term plumule has been mentioned. Plumule represents the shoot part, whereas the radicle represents the root part. So the embryo has been formed. And later on, when this grows more and more, it produces the seed. If you have broken a groundnut, you might have seen that there are two big cotyledons with a small embryo inside. So these are the two cotyledons in the process of development. And the small baby plant that you have seen in the groundnut, that is this embryo having the radicle and the plumule at the end. That is where the embryo development is taking place. But one thing to remember, this embryo, it grows bigger and bigger, consuming the nutrition from the endosperm present in the prime, that is present in the center of the embryo sac. The question commonly comes to your mind, is there any advantage of double fertilization? Yes, it has. The main advantage present in double fertilization of angiosperm is energy conservation, and the characteristic exceptional only to angiosperm. It produces a food supply for the seed. It provides extra protection for the species. It conserves energy. It allows for rapid seed development. So it does have certain advantage for the plant. After completion of these two topics, let us recapitulate. What is artificial hybridization? It is defined as a process of crossing over between two genetically different or individual having desirable trait to obtain offspring having superior trait than the parent. It can be achieved by emasculation in plants. What are the different steps of artificial hybridization? Selection, followed by evaluation, followed by emasculation, then bagging, tagging, pollination, and finally harvesting and storage of F1 seeds. What is the significance of artificial hybridization? It is the process by which only desired pollen grains are used for pollination and fertilization to get a target beneficial F1 generation hybrid. Let us understand and appreciate the process of double fertilization. In this process, two male gametes fuse with one female gamete, whereas the other male gamete fertilize the egg to produce zygote. The other fuses with the two pollen nuclei to form the endosperm. Two-time fertilization, thus the term double fertilization. Let us assess your knowledge just now. Instant assessment. I want you to try to answer this question. Fill in the blanks. E is defined as process of crossing two genetically different individuals having desirable trait to obtain an offspring having superior trait than the parent. Give a try. You have to give the terminology for that. Question 2. The selection of parent mainly depends upon the objectives of breeding program. True or false? You have to name the process by which stamen or anther of a flower is removed without affecting the female reproductive organ. You have to name the process. Two types of fusion, syngaby and triple fusion, take place in an embryo sac. You have to name the phenomena. A very tricky question, that in what type of plant emasculation is not required for crossbreeding experiment? Think, it is a very tricky question. Arrange the sequence of event in artificial hybridization experiment in correct sequence of event based on the steps. Bagging, emasculation, selection of parent and tagging. These sequences are in wrong order. The correct sequences may be there in A, B, C or D. You have to choose the correct sequence. You can take your time, try to solve it, and match your answer with the solutions which are given here. There are some takeaway home question. Define artificial hybridization. Mention the step involved in the process of artificial hybridization. Why emasculation is done in immature flowers. You have to expand PEN and PEC. You have to write the process of a double fertilization in angiosperm, and you have to mention why it is so called. Certain website and video links have been given. One H5P has been given to you for workout. Please copy this link or click on that so that when it comes out you can give it attempt and see the videos. This is a ZIT Bhubaneswar presentation. Keep